Thank you. Thank you very much. So thank you again for inviting me. And I'm very grateful to be able to talk to you. Okay, so let's get our things going here. So I have to first start by telling you a little bit about systems neuroscience and to see, how we can start talking as control theorists about this subject that's relatively new for us. So basically, this is a branch of neuroscience that studies regions of the brain, their connectivity, their behavior, and their collective behavior. So it's sitting at a higher level than neuroscience, which typically looks more at the neuronal level. There's very few people currently in control theory working in systems neuroscience, but one person is Jean-Jacques Slotine in MIT. He has really, I think he's a pioneer in this area, and he's obviously interested in sort of robotics implications of this subject. So for a control theorist who wants to get interested, who wants to have a look at systems neuroscience, I would say the core area that you want to look in the brain is the cerebellum, because the cerebellum seems to be the unit or the region of the brain where basically all of the behavior associated with the cerebellum is somehow related to control tasks. So the cerebellum sits right here. It's part of the small brain, along with the brainstem. It evolved 400 million years ago in sharks to help sharks visually track targets as well as reject sinusoidal disturbances from the tail of the shark. There's a cerebellum in almost all vertebrates. And the overall plan of the cerebellum seems to be the same in all vertebrates. You have here sort of a sketch of a cerebellum. And the cerebellum is divided into sort of major subdivisions according to its function. You have this outer part here, that's the ocular motor part of the cerebellum. This is called the vestibulocerebellum. It's also responsible for things like balance. And there's another little piece here, that's the vermis. So these two pieces are the vestibulocerebellum, which evolved earliest. So this is the part that evolved in sharks and then was copied by all other vertebrates. The spinocerebellum is dedicated to regulation of the limbs, arm movement, leg movement, and coordination of that movement. And to get an idea of how much real estate is dedicated to the spinocerebellum, you have here several homunculi, which show you how much real estate the cerebellum is dedicating to the various body parts. And this one you can see shows that the trunk is of little interest. There's very little going on in terms of control tasks for the trunk, but the feet are hugely important. The hand is hugely important. The head gets a lot of attention because there's a lot going on with the tongue, for example. There's a lot of fine motor control with the tongue, with speech. Also with head balancing. These are extremely important functions of the cerebellum. Finally, you have the last part of the cerebellum to evolve is the cerebrocerebellum. And this region is responsible for regulating cognitive functions such as attention and language and emotion regulation, other things like that. So what is remarkable about all of this is that each of these major subdivisions can be further divided into independent functional modules with a very sort of standardized plan. It's like strips or layers of functional modules, and each functional module has an identical microcircuit with identical input-output structure and identical circuitry. This means that if you can decode the computations of one of the functional modules, you then have a plan for how to decode the rest of them. It's said by the neuroscientists who worked on working out the cerebellar microcircuit, I'll list their names in a moment, that the cerebellum is performing a universal computation. And so the open problem in neuroscience is to know what is that universal computation. So these discoveries sort of culminated in work in the 1960s by Nobel Prize winner John Eccles and his collaborators. They mapped out the microcircuit of the cerebellum and identified all of the neurons and their connectivity. And they wrote a book sort of for almost like a popular reading with the title, The Cerebellum as a Neuronal Machine. They were really saying to the community, look, control theorists and computer scientists, we have here a very computational engine that's performing a universal computation. Come and have a look. There is a picture here showing sort of the folding structure of the cerebellum. The cerebellum is a highly folded brain region. It has more neurons than the entire rest of the central nervous system. And fortunately, like I've been saying, the neural circuit has been mapped out. So we can have a look here at a simpler picture of what that neural circuit is with respect to eye movement. So we have input signals. So the cerebellum, each functional module has two types of inputs. One of the input channels here is called the mossy fiber inputs. And this is gathering information from various parts of the body, depending on what functional module you're looking at. So in this case, we're looking at the oculomotor system. So it would be gathering information that's related to the oculomotor system. This information is being sent into a granule cell layer, which has a massive number of granule cells. And these cells are essentially filters. There's some kind of filtering going on there. It could be related to some kind of frequency decomposition. It could be related to some timing kind of processing, but, and there's many, theories about what this granule cell layer is doing, but it's obviously extremely important given the massive number of cells there. These cells then synapse on these parallel fibers. These parallel fibers then in turn synapse on something called the Purkinje cells. That synapse right there is known to be plastic. It's known to be adaptive. And the adaptation on the weights of these parallel fibers, some weights are being applied effectively through these synapses. And that is being mediated through the second input type of the cerebellum, which is called a climbing fiber input, which is a very, very different kind of input from these many mossy fiber inputs. There is only a single climbing fiber input for each Purkinje cell. When the climbing fiber fires, it causes a massive change in this synapse, and this is modulating the weights. Then the sole output of each functional module of the cerebellum is through these Purkinje cells to some kind of a hub, which is either, in this case for the oculomotor system, the vestibular nucleus, which is a collection of neurons, or something called the deep cerebellar nuclei. These are hubs that are collecting the output. The output is then sent out to contribute to whatever body part or system that's being regulated. So here we have the output going out to the motor neurons of the eyes that drive the muscles of the eyes and move the eyes. So you see this sort of closed circuit. Well, it's not quite a closed circuit, but you see this circuit with two input types, one output type. And then importantly, there is some extra piece of information we have, which is that each one of these output hubs, deep cerebellar nuclei, in addition to the vestibular nucleus, which is specifically for the vestibulocerebellum, each of these has a projection. Projection means connection back to the MF inputs in something that's called the nucleocortical pathway. And this is a very organized structure. Each functional module gets a feedback back to itself. So this is what's called in the neuroscience literature a recurrent loop. There's something that's recurrent, that's somehow needing to self-drive itself through this recurrent loop. So that's extremely intriguing. So if we step back and we just take sort of at face value these sort of high-level details and we write some equations, and this is, of course, a very naive effort, but we can start to sort of piece together what we're looking at. We can say, oh, there's some part of the body that needs to be regulated. This could be an abstract model of, emotion regulation. It could be the position of the eye. If we wanted to talk about limbs, we'd be looking at Euler-Lagrange models. We'd have to look at something more complex. We have a single sensory error that needs to be regulated. And we have disturbance and reference signals. And then we have a massive number of mossy fiber inputs coming in through some kind of filters, which we don't have a lot of information about, but some kind of filters. And we have a special input channel, which takes the output of the cerebellum from the Purkinje cells and sends it back into the input channel. So this is a special reserved filter precisely for that nucleocortical pathway. Then the parallel fibers gather up all of this information and form a regressor. This regressor is then weighted by some parameter weights, which are adjusted according to the climbing fiber input using a gradient law. Now, the gradient law is ubiquitous in neuroscience studies. You see it at all levels. It seems to be really a very solid concept of neurons are able to compute the gradient law, and it is believed that the synapse between parallel fibers and Purkinje cells is exactly this law. So I'm missing a transpose on this, this side. Then this output of the cerebellum is added to some other parts of the motor command that goes out to the system, and this completes our model. So this is a model that on a first look looks like an adaptive filter. This is the current hypothesis on the cerebellum, that the cerebellum is some kind of adaptive filter. And the current theory is that this adaptive filter is in fact a observer of the system that's being regulated. So the idea is that this here is supposed to be an observer for the plant. This is called in the neuroscience literature a forward model. So they use the term forward model for an observer. Okay, so we have come with, after our long period of reading on this problem, we have come with a different hypothesis. The idea is that we believe that behaviors such as visuomotor adaptation, which I will describe later in the talk, arm reach in a force field, if you have something pushing on your arm and you're trying to reach for something, walking in snow, standing in a lurching subway train, lifting a heavy object, reaching the arm in water with the vision distorted, making saccades to a moving target. Saccades are fast eye movements. Smoothly tracking a moving target with the eyes, holding the eyes in an eccentric position, suppression of interaction forces between limbs. These are all manifestations of disturbance rejection. This list roughly corresponds to some of the behaviors that are associated with the cerebellum. And so we hypothesize that the cerebellum is involved in all these behaviors of precision control precisely because it is the unique, centralized, and universal disturbance rejection unit of the brain at the behavioral level. There is, of course, disturbance rejection going on at the cellular level, at the neuronal level, but we're talking now at the systems level. How do disturbances get rejected at the systems level? We are hypothesizing that this is being done by the cerebellum. Furthermore, we observe that the cerebellum is performing this task of disturbance rejection with a paucity of sensory measurements. There really aren't many error measurements that the brain has access to because we have limited amounts of sensors. Although we have far more sensors, the body has far more sensors than any typical robot, still there is limited sensory information coming in, and yet somehow reference and disturbance signals are being rejected for smooth motor function and cognitive function. So this is, of course, for us, something quite familiar. This entire setup is you don't have full measurement of your disturbances. You don't have measurements of your reference signals. Well, we are and we're trying to regulate errors to zero for good function of our motor systems. This is regulator theory. And of course, our main tenet of regulator theory is the internal model principle. So one is led to the question, well, where is this internal model principle in the brain? Where is it? And so part of the dilemma, I should say, is that neuroscientists have not yet recognized that there is an internal model principle of control theory. This is one of our main challenges. Now, that doesn't mean that the internal model principle ideas have not percolated through that literature. They have percolated through that literature. You have already in the 1960s studies by great oculomotor researchers. So this is the oculomotor literature is fantastic because there have been really outstanding modelers in that area, outstanding control theorists have worked in that area. And so they have already experimentally verified that the human eye can track predictable moving targets, sinusoids, constant speed, and constant position with near zero steady state error, despite the fact that there is a very long processing time on the retinal error signal. It takes more than 100 milliseconds to generate that signal. And yet we have, as we all know, we have the human eye has near perfect tracking capability for predictable targets. So that is remarkable. So we know that there has to be an internal model somewhere. You have then, in this time frame of the 90s, you have people writing papers about internal models of gravity in the brain and of linear acceleration, or does the brain model Newton's laws? Well, that has a natural analogy with, does the brain have a notion of an exosystem? And you have papers here again, like internal models of the physical laws of motion. There then comes in 2009 kind of a crystallization with what I consider a very important experiment by this team, Chairman Ara, who do an experiment where they have a monkey tracking a constant speed target. And during this eye tracking task, at some point for, let's say, 1 second, experimental device called an error clamp is introduced. And what this means is that rather than having the eye move based on the target is moved based on the eye. So the experimenter manipulates the target motion so that it's centered on the fovea. So for this 1 second period of time, they then record inside the monkey brain, and they see in the cerebellum is firing according to the motion of a constant speed target, despite the fact that there is no error signal, the error is zero, and in some sense, the target has been made moot by the fact that the target is now moving according to the eye. So they postulate that it must be that the cerebellum has a model of this visual target that continues to drive the tracking of the eye without actually a real target there. Also in 2009, this researcher, Liz Berger, who has written many wonderful experimental papers, he wrote a summary paper, a little bit almost like a survey of his career. And he basically had 3 hypotheses on what's going on in this region of the cerebellum that's dedicated to eye movement. He said, theory one, the cerebellum has a model of the motion of external objects. 2, the cerebellum has a model of the oculomotor plant. That's the forward model. And 3, the cerebellum has a model of the oculomotor plant plus head movement. So it's the first hypothesis that corresponds for us to the internal model principle, basically a model of the exogenous of what's outside in the world, as opposed to an observer. What then happens after that is that, unfortunately, it seems like there's a sort of a convergence of thinking here in these papers in 2009. But really, the idea of the internal model principle of control theory is somehow dissipated. The concept is not known in neuroscience, which is remarkable. And then you have, now you have some recent papers where now the theory is that, the cerebellum is involved in active versus passive self-motion sensation, or, it's some perceptual task that's going on. So it's still sort of related to sensing and exogenous behavior, but you don't see a crystallization. You have a cacophony. And this is happening, meanwhile, while there's many, many people who are dedicated in neuroscience to the forward model theory. And there are careers that have been made basically over 35 years of experiments where this is the theory they're working on. So this is the state of affairs today in neuroscience. So then on the control theory side, then we can say, well, what does control theory have to offer here? And this is a little bit where I jumped in, right? So I was reading all of this literature, looking at the state of affairs, and I was just truly astounded that the internal model principle of control theory had not been identified as a key component of some part of the brain's behavior. So I started working on this hypothesis of the cerebellum has adaptive internal models. So the setup, to give a little overview of what's the setup on adaptive internal models, in case you haven't looked at this literature, is that you have, let's say, take a simple linear time invariant system. It's already been transformed according to the regulator equation. So we have a matched disturbance matched to the input. We have an error that we want to drive to zero. It's single input, single output. And so, of course, the task is to reject the disturbance, which can include also tracking objectives. We assume that the disturbance is modeled by a linear exosystem, and this might not look familiar if you're thinking of how you learn about exosystems in your undergraduate or graduate courses. You're used to seeing W dot equals SW. This parameterization of the exosystem is very nice because it sets it up for adaptive control. This is something due to Nikiforov back in the early 2000s, I think, 1997, he has a paper, and he writes this very beautiful parameterization with FG, a controllable pair. And the way to understand this is that if you plug in this D into here, you get a system W dot equals F plus G psi transpose W. The F plus G psi transpose is the S that you're used to. So this parameter psi is sort of a minimal parameter set to set the eigenvalues of the exosystem. But it's also in a form that's ready for adaptive control. So that's a neat innovation. Now, in the adaptive internal model literature, you have at this point, starting from around 1996, with some of the early work of Nikiforov, up until, you have the contributions from Sirani, Isidori, Marconi, Marino, Tomei. You have many, many players in this area. There are many designs now for adaptive internal models. So I have, you can have states measurable, error feedback, plant known, plant unknown. You can have time delays, you can have state derivative measurements. Most of these results there's sort of one key limitation in this work, is that mostly it's focused on linear systems, I should say. There is some work of Isidori on nonlinear systems, but the main sort of overriding assumption that you tend to find is a minimum phase assumption on the plant. So that's maybe there's still some frontiers there of research that has to be done. But here's an example of a typical adaptive internal model where X is measurable and the plant parameters are known. That's not realistic in a biological setting, but we do this for the purposes of pedagogy. This is basically a Kreisel-Meyer filter, a Kreisel-Meyer observer that takes the pieces of the right-hand side and reconstructs them so that you can get your hands on the disturbance. So here is the regressor W hat. If you write the derivative, if you write the dynamics of W hat, you will recover these dynamics. So you get W hat dot equals F W hat plus GD.
If you then write the estimation error W tilde as W hat minus W, you will get W tilde dot is F W tilde. F is Hurwitz, so you get that W hat is converging exponentially to W. So this is a proxy for the regressor. Then here's a parameter adaptation law that you add to try to get your hands on this psi parameter, which again is encoding the frequency content of the exosystem. So this is basically trying to match the frequency content in your disturbance. And I'm using sort of a standard, this is supposed to represent the error signal. And here is then the output of the internal model, and you combine that with some stabilization piece in case this A is not already Hurwitz, and that's the full design. Notice that this filter, importantly, this filter right here is the one that provides the internal model principle, because if you substitute the U in here, you will get a system matrix here. When the psi hat has converged to psi, you will get a system matrix on this filter, which has the eigenvalues set at the eigenvalues of the exosystem. So there you go. The internal model principle is right there. Now, this is what control theory has to say. And now we can see the resemblance to the adaptive filter model of the cerebellum. We can see also the very important role played by the nucleocortical pathway, that recurrent loop. That is, if we make the analogy with this internal model, that is the pathway that when it closes, you get an internal model. And that's why would the brain have decided to make a special pathway out of the output of the cerebellum back into the input? Well, I have a conjecture that is just purely my conjecture, but my conjecture is that you want to turn on internal models in the brain, and these are unstable systems, right? We're tracking sinusoids and ramps and constant. So we have an object, an exosystem that's not stable, that's neutrally stable, and we want this to happen in the brain. Well, you can imagine that you're going to have to be able to quickly latch on, off these internal models as they're needed. And this nucleocortical pathway gives a nice, clean architecture. You cut that link, cut this link, and this is Hurwitz, so you're stable. So it makes sense. Of course, it's all, this is extemporizing from the math, and we need our neuroscientists to come in and say, can this be real? Can this really be the explanation for this very particular architecture? Okay, let me check my time. All right, so now I'm going to tell you a little bit about the oculomotor system. We felt that it was hard to know, like, now what? So we think, we make this hypothesis and we think this is what the cerebellum is doing, but now what do we do? How can we plausibly make these claims just based on these wonderful mathematical analogies? My feeling was when I started in this, well, I'm going to have to give some evidence, and the oculomotor system was the right place to start because the neural circuit is reasonably well understood. There are hundreds of experiments in the literature just on this system. And I thought, okay, let's use our idea and let's see if we can produce a model of this system, this one system, and then maybe we have something real that we can offer as evidence. So the oculomotor system, basically its task is to stabilize objects on the fovea. There's also more generally a task to ensure that the visual field doesn't move too fast. So when you move your head very fast, your eyes will actually swing the other way. You're not aware of that generally, or maybe you are, but generally you wouldn't be thinking about that because it's a reflex, so it's automatic. You don't have to think about moving your eyes. That's, in fact, the vestibular ocular reflex. So there's a great deal of emphasis on keeping the visual field stable, because if the visual field moves too fast, you cannot see. And if you are a shark or if you are a fish,
and you can't see, obviously you're not going to survive as a species. The oculomotor system is divided into several subsystems, and we can classify the blue ones here as the so-called slow eye movement systems. Then we have a very interesting system, the saccadic system, which is a very fast system. It is what lets you quickly look at different objects in your visual field. That needs an entirely different kind of modeling approach than this one. And there's also a vergent system that keeps the two eyes sort of in track. So the pathway in the brain that we're looking at for the blue system, which is what I'm going to discuss, this is the slow eye movement system, is starting from the visual cortex, moving down through this pontine nucleus and sending signals to the cerebellum, which is this floccular complex. This is the visual part of the cerebellum. Taking its output, sending it to one of these nuclei, deep cerebellar nuclei, or in this case, it's called the vestibular nucleus, which is in the brainstem, and then sending out commands to the motoneurons that drive the muscles of the eyes. And then there's another pathway here that is gathering up and collecting, preparing that climbing fiber input, which is a sensory error signal that I'll show you in a second. What that is. So one of the challenges we face when we work in this area is that we have a neural circuit. So this is a very high level schematic of a neural circuit. And we have to, when we build models, we have to abide by the measurement structure that is imposed by this neural circuit. That's, I think is not surprising. Control theorists are very, very comfortable with dealing with circuits. In general, that's a paradigm we love. So to me, that's not a make or break, but some aspects of control design are a little bit finicky when you have to work with a neural circuit. So to model the oculomotor system, the basic thing, the key message that you need to know is sort of just this picture. There's a target in the visual field. It enters through the lens and it projects onto the back of the retina. And this target here can be identified by the brain, and the distance along the retina from the fovea is available to the brain, and that's the retinal error signal. This angle is approximately the same as the angle in the visual field, assuming this target is far enough away, which most targets are far enough away. So that you can say that this signal here, this distance here is proportional to that angle. The oculomotor plant, you can model it as, we're modeling here a single human eye, just horizontal motion. You can model it with a first order model. There's also a second order model, and you can also use nonlinear models. We took the very simplest model, which is this simple first order model, where X is the eye angle. And we also need to know about the X is the eye angle with respect to the head, and then the head angle is in a world frame. So we have a scalar plant, basically, which is stable. And then we have a retinal error signal, and the entire objective of the cerebellum and of the oculomotor system is to drive the error to zero, whether for tracking objects, whether for looking at fixed objects, or whether to stabilize the visual field. A small note that there is no proprioception of eye position in the brain. The brain does not have sensors in the eyes to tell you where the eye is looking. So there isn't such a function. So the only measurement the brain has directly is this error signal. So I've skipped sort of through further development to sort of just give you directly what the model is. So we have our oculomotor plant, first order. There is in the brainstem a brainstem neural integrator, and it appears from all of the experiments that that is providing an estimate of the eye position. So there is that piece of the model. Here is an adaptive internal model. So this is modeling the part from the cerebellum. And here I'm using a little bit of a different design. So I continuously switch between adaptive internal models because it's sort of like a plug or play with off the shelf design. So this is the design of Serrani, Isidori, Marconi from the 2000 or 2001 paper. So it's a little bit simpler in structure, and it's works works very well for getting a simple model. The brain heavily utilizes reflex signals. So we have here a brainstem signal that's got basically a direct feed through of a head velocity signal, which comes from the ears. And that is providing you the VOR. It's moving your eyes opposite to the head velocity. And there is another reflex signal, which I will not discuss too much, but it's basically what this thing is doing is it's trying to take away some of this drift. This damping term means that the motor command constantly has to overcome the pulling of the eyes back to center. And so this is facilitating making the eyes seem like they're more easily, like they're pure integrators, that they just glide around. There is a visual signal that is stabilizing. This is developed in the visual cortex and comes down to the brainstem. And then finally, there's the output of the cerebellum. So it's a quite simple model, but innovative. Hasn't been tried before. And it turned out to be something of a watershed. Here's the architecture. We have our plant, the oculomotor plant. It generates the state, which is the eye position. Comparing to the target position and the head position, you get an error, which goes in as the climbing fiber input to the cerebellum. Here is that recurrent loop, which we see present in our model here, because this is going back into the cerebellum. Okay, sorry. Okay. The brainstem is the hub. It's gathering up all the signals for stabilization. It's taking the signal from the ears and generating that reflex, the VOR, and then combining with the cerebellar output and forming the motor command. So this is the architecture, which is consistent with the brain's architecture. So that's important. Then we ran a battery of experiments with stimulation. So we went into the literature and we pulled out roughly 30 experiments, which the details here don't matter so much. I will highlight just a few. But when you go into this literature, you find three types of experiments. You have behavioral experiments where you have a monkey or a human just performing some behavior like tracking a sum of sinusoids. You have neurological experiments where they're actually probing the brain and measuring the spiking behavior of individual neurons, typically on the output of the cerebellum. So that's usually done with monkeys, well, always with monkeys, or with rabbits or different animals. And then you have lesion experiments where neuroscientists will either induce a lesion in an animal, or you have a patient who has a lesion because of a stroke. So there is a part of the brain that's not functioning, and that helps you further test your model. So this gives you a battery of experiments that you can try out, which we did. And what basically happened is that our model matched all of the experiments. We found, like I say, around 30 experiments, and there is no model of the oculomotor system that's comparable. Many of the models don't have the cerebellum, and that's the basic problem. But even the models that do attempt to model the cerebellum, and typically will put a forward model in the cerebellum, they can only recover certain behaviors and not others. And so it is really like there's a watershed here in the sense that there's something going on, something we have to look at. Speaking about the 2009 experiment by Cherminara, here is that experiment where the eye is tracking a constant speed target, and from 5 to 6 seconds, the error is clamped. And that is not easy to see here, but during this 1 second interval, the error is exactly 0. And the eye, we see that in the model, mathematically, the internal model, of course, continues generating a ramp. And so the eye continues to track. Then when the true error is generated again, of course, there's some transient. And this shows the velocity of the eye.
These are experiments with tracking sinusoids. This is showing even the transients are matching. This is showing that even with a 100 millisecond delay in the retinal error signal, we still have good behavior. Okay. We then went on and we said, okay, let's have a look at discrete time behavior. And let me check my timing. Okay. So we wanted to also study visual motor adaptation. So this is also a very interesting, this is fascinating problem of a behavior that's truly discrete time. You have to think about a baseball player who's trying to hit a ball, but the baseball player is wearing some goggles that, let's say, shift the vision down. So the baseball player sees the ball coming and the ball is lower than he thinks. And so he tries to hit lower. And of course he misses the ball because the ball is actually higher because the goggles are wrong. So through repetitive trials or very fast swings, the baseball player will gradually adjust his movement so that the bat will be higher, even though what's seen is lower. So this is maybe a little bit hard to get your head around, but what this is basically doing, this is a system of the body that is calibrating how we move relative to what we see. If you think about it, that's not obvious. It's not obvious that my motor command to reach my hand out is going to go right to where I see. I need to calibrate that. And if I wear magnifying glasses, that's going to change it. If I wear glasses that invert the world, that changes it. And so this appears to be a brain process that's described as machine-like. It has a very, very characteristic imprint. Taking place over repetitive trials with a visual error closely following the movement. That's important. Within a say a 300 millisecond window, you have to have an error measurement. Boom, you get that error measurement, and that's a trigger for a kind of discrete update. And through this repetitive process, you are calibrating essentially the mapping between what is seen and how to move. And this is fast movement. This is not slow tracking, but this is fast movement. So we decided to model this as we took a single degree of freedom. We were inspired by an experiment with fast arm reaches, where people make reaches to a target, but they are shown not their true hand position. They're shown a cursor on a screen, which is rotated. So the single degree of freedom now is the angle of the arm in that experiment. So we took a single degree of freedom. So we have a scalar model of the plant, and we're trying to calibrate the position of the hand in this case so that we can reach a target in the visual field. So we have a visual error, and we have repeated trials, so it's a discrete time model. We made some simplifying assumptions. There's no proprioception, so you have absolutely no measurement of your hand. You're moving your hand too fast. There's no way to measure where your hand is. The only measurement you have is an error. And we also assume that there is a disturbance, which in this course corresponds to the subject being presented a cursor position on a screen that's rotated. And we made our key assumption that that disturbance is predictable, can be modeled by a linear exosystem. So we write again our open loop model. We propose yet another variant of an adaptive internal model, which is here, but it has very similar structure. And the only difference here is that you don't see a parameter adaptation process, because the experimental evidence seems to be that the adaptation is very slow. So we have basically chosen a fixed parameter psi here. And what we found out is that in this case, we also recover the main behaviors of this system, this visual motor system. And these have been cataloged in this wonderful paper by Smith in this 2006 work. And so there are these prototypical behaviors, and you may have heard these behaviors when people talk about learning: savings, reduced savings, anterograde interference. These words are all characterizing transient behavior of a learning process, in particular this visual motor adaptation.
So transients are generated by switching the value of the disturbance. So here's an experiment where you have a disturbance of, I think it's minus, the yellow is the disturbance. So it's, I think, minus 30 degrees. It then pops up to plus 30 degrees, and then it's minus 30 degrees again. And with this sort of paradigm, what you notice is that the hand has to go opposite to the disturbance in order to hit the target at 0. So the hand is approaching 30 degrees. And you can see this nice asymptotic behavior that you would expect of a typical control loop. And savings, it refers to the fact that the second time you do this experiment, so the second set of trials where you have minus 30 degree disturbance, it's faster, which is shown in this picture. You can learn faster. Why is that? That is because the cerebellum has built up an estimate of the disturbance. So once you have stored that estimate, then you have a very quick counter movement with an opposite disturbance. But once you go back, the cerebellum still has remembered that, I have a minus 30 degree disturbance, and that comes back very quickly. So that's the explanation of how you get all of these behaviors that are all sort of transient behaviors can be explained in terms of thinking of the cerebellum in terms of a estimate of the disturbance in visual motor adaptation. Okay, so I will try to now kind of wrap up a little bit. There are a number of lessons learned and challenges in our work, and I've mentioned some of them. These are challenges that I see for control theory, I should say. I've mentioned neural circuit constraints, the fact that in some sense, control theory, I think, has two extremes, either fully decentralized or fully centralized. And so working with neural circuits has some strange things, like you can say, we have a state measurement that's available to the internal model, but we can't use it for stabilization. That's a strange way of thinking for a control theorist, but these are the types of constraints you get when you have a neural circuit. So it's a little bit different than just decentralized control. I've talked a little bit about the issue of non-persistence of error signals. We tend to think of a control loop as something where you have measurements that are sort of perpetual, but in the brain, error signals are coming on and off. And so there needs to be a technology to quickly disable internal models. That appears not to have been studied in the switched systems literature or in the regulator theory literature. A very interesting area, I think, is the area of optimal steady state. Optimal control tends to focus on optimizing the transient, how you go from A to B. But we've noticed in our work that the brain is putting out quite a bit of, is building up infrastructure to deal with steady state costs. And so a control theory that optimizes the steady state is a little bit not present. It's not present in sort of current thinking. The other thing we notice is the ubiquity of feed-forward reflex signals, which if you look at our typical robot design, it's not there. We don't use reflexes in robots, but we should. The brain is heavily utilizing reflex signals to cancel brief disturbances. In other words, to say the mantra that we have as control theorists, that if the system is GES, then it's robust. The brain is not happy with that at all. It is interested in instantaneously canceling brief disturbances whenever it can, if it has the sensory measurements. There's also another issue that brain signals are very noisy. And so it appears that the brain is having to use slow adaptation processes to average noise. Whereas in control theory, we often get stuck. I find when I read the literature, you often see that people are really stuck in making the transients better, making it faster. We have, backstepping design. Let's make it faster. Whereas in the brain, that may not be the concern whatsoever. It may be entirely a different issue. And finally, the last thing hugely important, is an entire area on its own, is that adaptation in the brain is robust, even when models are over-parameterized. Okay. So I have one last part, and I don't know what my timeline is, when you would like me to quit.
Well, you're doing fine, Mireille. So if you're winding up in the next 5 minutes, then there's typically 10 minutes for questions, and
okay.
The speaker and the audience.
Okay, well, if you tolerate another 5 minutes, it's going to be fast, but I'm going to squeeze more in. Okay. All right. What I've done so far, some of you may think, well, gosh, you know, you're making things nice for systems neuroscience, but what do you have to say for control theory? My view is that if we go and make contributions to systems neuroscience, we will, as control theorists, there will be a windfall for us too. So let's ask a converse question, not what can control theory do for systems neuroscience, but what can systems neuroscience do for control theory? And I'll take the very last topic, key issue in adaptive control, overparameterization and lack of robustness. So let's talk quickly about robust parameter adaptation. So if you study adaptive control, you know that adaptive control nicely lays out in terms of error models. So the setup is you have a measurable regressor, you have some unknown parameters, you have a parameter estimation error, and two standard error models are the static error model, which is very well known, and another very well known model is the dynamic error model for linear systems. There are also very famous and beautiful results on persistence of excitation of the regressor, gives you results on global exponential stability of these models, in addition to some other conditions on the system here. And then in turn, from global exponential stability, we get robustness to bounded disturbances. And that's the way we normally get robustness in adaptive control. The problem is that if W is not persistently exciting, then it's famously known through some famous counterexamples that these models are not robust. So this led to a great deal of research in the 1980s, of which Peter Keynes is a contributor, with modifications, so-called modifications of the standard adaptation law here, which is the gradient law or least squares. These are called the sigma modification, the E1 modification, dead zone projection. You can read this beautiful book of Ioannou and Son, which really lays it all out for you. It's a fantastic book. More recently, people have been looking at how do you get rid of the PE assumption. And what they're trying to do is extract more information out of the regressor. So even if the regressor is not persistently exciting, they're doing filtering or collecting data points from the past or using some initial transients to get more information out. That is the trend in control theory right now. And there's a lot of excitement because there's a lot of new methods that are coming online that will sort of like resolve this fundamental obstacle of adaptive control appears to be on the cusp of being resolved. Now we look at neuroscience. How can neuroscience inform us? And we see a totally different kind of perspective from the point of view of what the brain does. The brain operates according to the use it or lose it principle of neuroplasticity, which if we put it in terms of our mathematical terms, if a parameter estimate is not excited by the regressor, then it should be gradually forgotten. Unwitting overparameterization should have no deleterious effect on regulation or robustness. That's what the brain thinks. Okay, that's a different way of saying it. What can we do with this idea? So here, let's take a look at one idea we have. So suppose that our exosystem is generated by an suppose our regressor is generated by an LTI exosystem. So we're dealing, with disturbance rejection type problems. If our regressor is not PE, then it is known that you can write this regressor as the product of a full column rank orthogonal matrix times a persistently exciting regressor. Now we perform a coordinate transformation using a orthogonal completion of this orthogonal of this matrix with orthogonal columns to form this orthogonal matrix. So we make this coordinate transformation of our original parameters to these parameters. We call these the PE and the PERP parameters. If you look at error model 1, the static error model, then you see that you have a nice splitting of your parameters. You have a piece here that is the classic static error model, but now you have a regressor that's persistently exciting. Instead, you have a piece down here that's not asymptotically stable. And this is precisely the problem with parameter adaptation, is that this piece is not robust. So you have a GES piece and a not robust piece. So then now let's take one of the typical modifications, the sigma modification, which I believe was due to Kokotovich and Ioannou. It basically adds a damping term to try to overcome this problem here. If you then transform to our coordinates, our new coordinates, you see that, yes, we resolved the problem of lack of robustness by making this exponentially stable. But we have introduced this term, which means that this system will no longer converge to the true parameter values. And that means that the error will not go to 0. In fact, these parameters will not even go to equilibrium because this is time varying and this is supposed to go to a constant, and it doesn't. So you can see very clearly that in the methods of the 1980s, you find this trade-off between regulation and robustness. We introduced a different method based on the use it or lose it principle of neuroscience. We say, let's put in a different modification. It's like the sigma modification, but it's a little smarter. So it has this term here. And what happens? We apply our coordinate transformation. And I didn't write the error equation, but we have our original exponentially stable static error model. And we have our exponentially stable parameters. These are the parameters that are being forgotten. These are the parameters that are not excited by the regressor. And now we achieve global exponential stability, but we also achieve regulation. So this is some new work we have with my student, Eric, just accepted by systems control letters. And of course, what I've shown you is an idealized case where somehow this information is available. Of course, this information is not available. And so what we need to do is develop methods to estimate this projection map. So this has been a little story about the fact that, okay, one area can inform the other, but also systems neuroscience can inform control theory. So in conclusion, a Newtonian revolution is underway in systems neuroscience. Control theory will play a major role. Just as physics has enriched and informed the development of mathematics, systems neuroscience will enrich control theory. Our current projects, we're developing a control architecture for reflex adaptation. We're developing a framework for optimal steady state regulation. We are continuing our work on robust parameter adaptation, and we have a project with some neuroscientists under a New Frontiers project on a further generation of our sensory motor adaptation model. So thank you very much for listening.